Thank you for having me, and thank you for introducing me. My work focuses on this broad class of diseases called fibrotic diseases. These diseases can affect really any major organ, and the underlying disease can really vary depending on the organ. But the central kind of mechanism, pathophysiological mechanism for this disease is accumulation of collagen-rich scar tissue at the organ. And this inhibits organ function or messes with organ function, and it eventually leads to organ failure. Now, these diseases are a huge burden, and they're estimated to account for about a third of deaths in the industrialized world. So there's been a major efforts in developing pharmaceuticals that can reverse fibrosis, because that's really the issue at the heart of these diseases, that once that scar tissue forms, we can't degrade it. But despite these major efforts, we really haven't been successful. And that's because we don't really understand the biochemical factors that dictate scar degradation. So my lab is uh, investigating these biochemical mechanisms that dictate scar tissue. And we started in the heart, because in the heart, you can have an injury like a myocardial infarction or heart attack, where a major part of the myocardium dies. So the cells die after this injury, and then the body starts to do what the body does with any wound. It repairs that by removing those dead cells and laying down scar tissue. So the scar tissue development is good because you don't have a hole in your heart, but on the other hand, the scar tissue can't do the same thing that the heart does. The cardiomyocytes, the scar tissue can't pump, it can't function the same as the organ, and so that eventually actually leads to increased remodeling and eventually heart failure. And this is true for all adult mammals, but there are organisms like the adult zebrafish in which the same type of scar injury also results in development of scar tissue. But unlike us, the zebrafish can completely remove that scar as the myocardium regenerates. And in both cases, that scar tissue is made up of the same stuff. It's collagen rich, and that's what gives structure to the scar. Despite the similarity, in one case, the scar is permanent, and in the other case, the scar is resorbed. And the dominant theory in the literature had been that the degree of collagen cross-linking is what dictates whether or not scar is reversible. And those collagen cross-links are the bonds between the collagen molecules in scar tissue that gives it stability. So we set out to answer this question: is degradable collagen less cross-linked? And to do this, we looked at the biochemistry of collagen. We know quite a lot about. We know collagen's cross-linking starts with the oxidation of collagen lysine groups to form these al-lysine aldehydes. These aldehydes are key because that's what initiates cross-linking. They can react with each other or with other lysine groups on nearby collagens to form these early cross-links, and those cross-links can further react and rearrange to form the stable scar tissue. But as I mentioned, the key here is formation of these aldehydes. They're the only enzymatic step in the collagen cross-linking cascade. And as chemists, we love aldehydes because we can target them very well and very specifically. So we develop probes that bind to these aldehydes and accumulate on oxidized collagen, thereby giving us an idea about the degree of collagen cross-linking that's going on. We can really use any imaging modality, but in this case, because we're looking at the zebrafish heart, we used fluorescence because we needed the high spatial resolution. So of course, I don't have time to tell you about the development of these probes, but suffice to say, we do a lot of development and validation of our probes before we use them. And then we use the optimal construct, which in this case is called TMRO, in these subsequent studies. So something I should mention is that our probes can also bind to healthy tissue that's rich in oxidized collagen. So for instance, the mammalian aorta, it's not part of the heart, it's the tube that takes blood from the heart to the rest of the body. But this tissue is rich in elastin and collagen, and so our probe always binds there. And this we use as kind of a positive internal control. So the story here was that in the mice, what we saw is that at early stages after the infarction or the injury, we don't see much accumulation in the ventricle. But that changes quickly, and starting at 10 days after injury, we see robust accumulation of our probe at the injury site. We can quantify this, and we see that it peaks around 21 days after the injury. and then it decreases after that. But that's not because the scar is decreasing. We still see scar tissue. So there's areas of scar tissue that's clearly scar tissue, but the probe doesn't accumulate there because that scar is mature. The aldehydes have already been consumed. So in the mice, we see that collagen cross-linking is robust even 2 months after injury, and there is evidence for scar tissue maturation. So in the zebrafish, we use the line of zebrafish that has GFP, cardiomyocyte-specific expression of GFP, and that allows us to readily see where the area of injury is. And then, as I mentioned, we always see our probe accumulating in the structure in the zebrafish is called the bulbus, but it's very similar in structure and function, composition and function to the aorta. So the story in the zebrafish was actually kind of the same. We don't see much accumulation of the probe in the ventricle at early days after injury, but that changes pretty quickly. And similar to mice, we see that cross-linking peaks around 21 days after injury. But this is despite the fact that the injury volume is decreasing, so regeneration is happening. Then at 62 days after injury, the myocardium is completely regenerated. There's no longer scar tissue in the ventricle, and we don't see any accumulation of probe. So what this showed us is that despite extensive collagen cross-linking, the collagen is still being degraded and resorbed. So really, if we compare collagen cross-linking these two species at its height at 21 days after injury, there's really not much of a difference between the zebrafish and mouse collagen cross-linking in the infarct. And that led us to look at what happens to those cross-links. What's the fate of those early cross-links? Do they mature in both species? What we looked at was this degradation-resistant collagen cross-link that we call PYD. And when we quantified PYD on collagen in the infarcts, we see that it's highly abundant in the mouse tissue, but we really don't see it at all in the zebrafish tissue. And it turns out that this is because there's a hydroxylation step that needs to happen before PYD can be formed. So collagen can be hydroxylated before cross-linking, and this by this enzyme called lysyl hydroxylase. And whether or not collagen is hydroxylated, it can undergo cross-linking. So it will still get oxidized no matter if it's hydroxylated or not. But if it is hydroxylated, then that will allow formation of these PYD cross-links. And so when we looked at the degree of hydroxylation of collagen, we saw that the mice have a highly hydroxylated collagen, but the zebrafish does not. And so it turns out that these mature collagen cross-links are absent in the zebrafish infarcts, and that's because collagen in zebrafish infarct is less hydroxylated. So if you take one thing away from this talk, it's that it's not the degree of collagen cross-linking that dictates scar tissue resorption. It's really the type of collagen cross-link. And the collagen in the permanent scar of the mouse is locked into place, whereas in the zebrafish, it's cross-linked, but those cross-links are open. And so we're now pursuing this as a potential therapeutic target where we're trying to inhibit formation of those locks without interfering with normal wound healing. So that's an area of research that I'm exploring now. So then the last couple of minutes, I want to tell you about our efforts in translating some of these technologies. We're interested in developing these technologies to be able to stratify patients. So in a lot of fibrotic diseases like idiopathic pulmonary fibrosis, the patient population is very heterogeneous in terms of disease progression. So you have some patients that progress very rapidly, or some patients that are progress very slowly, or anywhere in between. And the problem is that we have no way to distinguish between these patients. And when they're enrolled in clinical trials for treatment, they're all lumped together. So that ends up diluting any effect that you would see with treatment. And so what happens as a result is that the majority of clinical trials fail to show drug efficacy. So we are developing these tools to look at disease activity, which would help us stratify these patients and improve clinical trials. So we target the same sort of collagen cross-linking cascade. And again, the key here is that these aldehydes are the only enzymatic step in collagen cross-linking, and they're only expressed during active disease. So these aldehydes are not a part of stable scar tissue, and therefore targeting them would enable us to quantify disease activity. And in this case, we developed probes that have a radioactive gallium-68 that allows for PET imaging, but this works in the same way as the probes that I showed you earlier, where you get binding of these probes to the lysine aldehydes on collagen, and therefore it gives us a readout for the degree of collagen cross-linking or activity in the tissue of interest. And in this case, what I've shown is that in a mouse model of lung fibrosis, our probes can really sensitively detect changes in fibrogenesis with disease progression. And this is something that the state-of-the-art imaging technology, high-resolution CT, cannot do. And so we really think these lysine binding probes can non-invasively track fibrogenesis and distinguish active disease from stable scar, which will allow us to stratify patients based on disease activity. So our lab really is developing tools that enable understanding disease mechanisms, identifying therapeutic targets, and de-risking clinical trials eventually. So with that, I'd just like to thank everybody that I work with, including my mentor, David Sosnovic and Peter Caravan, and our collaborators, and also my sources of funding. And please feel free to reach out to me with any questions or comments. Thank you.